cloud. Welcome and thank you everybody for coming today. My name is Darcy Dugan and I'm with the Alaska Ocean Observing System and the director of the Alaska Ocean Acidification Network. And I'm joining today from the traditional homeland of the Dena'ina people where I live in Anchorage. And I'm really excited to see you all participating. I think we are gathering here because we are interested in ocean acidification and how it's going to affect Alaska. And some of us are curious and some of us are concerned, and probably all of us are, or most of us are curious and concerned. And I think this is going to be a really nice opportunity for us to learn from one another, both what is going on with the latest science and what we know about our waters, and also how we're responding to this science and ideas of how we can apply what we know so far in relevant ways. So today is the first of a 4-part discussion series that's being organized by the network. And I just wanted to give a brief background on the Ocean Acidification Network. It started in 2016 with the goal of trying to expand our understanding of ocean acidification, both processes and consequences, and also look into potential adaptation and mitigation measures. And some of the main roles that we play are helping scientists collaborate, trying to identify gaps in research and monitoring, and then engage with Alaskans to get everybody up to speed on this issue, and also understand the information needs around the state and how we can best apply what we're learning so far. So we knew at the beginning that Alaska was going to be a hotspot for what is a global problem, and it was going to take a collaborative effort to try to figure out what's going on and what it would mean for people here. And we started out with about 12 researchers, and it's exciting to say that the number of people that are doing work on ocean acidification in Alaska has more than tripled since that time. So new scientists, both in the oceanography and species response side, there's new labs, there's new grad students, and there's a lot of other marine scientists who have agreed to add sensors to their projects or collaborate on interdisciplinary research. So we have 44,000 miles of coastline and a huge dependence on marine resources. So it's a good thing to see this community growing. But I also want to emphasize that one of the most important parts of the network is that it's not just researchers. We have a really vibrant ocean acidification community that we're looking at right now across this screen. So it's engaged fishermen and fishing organizations, it's tribes, it's community samplers, it's educators, it's concerned coastal citizens, and it's going to take all of us. So we are really glad that you are part of this community, and we're grateful for your commitment and your energy here. And I want to just give a preview of what we have before us. The Ocean Acidification Network Executive Committee started thinking about the idea of a discussion series back in November. And we wanted to design it in a way that would be most responsive to people's interest and needs. So we circulated a survey that went out to the network listserv and beyond, and about 100 people responded. And we put together topics based on what we heard from you. So over 4 sessions, we'll be addressing regional conditions. So what we know about carbon chemistry around the state, that one's happening right now. Species response, so what does ocean acidification mean for mariculture and subsistence, and also what does it mean for commercially harvested species, so groundfish, salmon, and crab. And then the 4th one will look at adaptation and mitigation. So how can carbon dioxide removal, carbon sequestration, and other natural climate solutions help us to adapt or mitigate ocean acidification? So these sessions will run every other week through early May. And what we also heard back from you is you primarily wanted to come to learn as a first priority, but also important was spurring collaboration on research and action, and also documenting key issues and needs so that we can communicate that to funding sources, the research community, and also policymakers. So our approach with these sessions is to bring in subject area experts to provide an overview on these topics, and then provide time for structured conversation in smaller groups. So a very important part of this series will be two-way dialogue. We want to make sure that we are sharing the latest knowledge, but also that the research community hears what's on the minds of Alaskans. So we're hoping this will bring us together in some new ways. I want to introduce Molly Mayo with the Meridian Institute, who will be our facilitator today. And she's been doing facilitation and mediation work focusing on natural resource management and policy for over 25 years. And she's going to kind of line out how today is structured. But before that, I just wanted to give a shout out to the Ocean Acidification Network's Executive Committee. I think Molly's going to put their names and affiliations in the chat, but it's Amanda Kelly, Tom Hurst, Jamie Gaughan, Scott Goodman, Chris Holderreed, Jeff Hetrick, Dorothy Childers, and Jessica Cross, who have put a lot of time and energy into thinking about how to structure and implement these sessions. And I guess one more thing before handing it off to Molly, I have a question that's going to go out in poll form to the audience. I'm going to hit a launch on this. We have a question for you about your level of understanding of ocean acidification. So we just have a sense of audience here that will help us as we go into the discussion. So if you take a sec and fill that out, I'll now hand it off to Molly Mayo.
Thanks, Darcy, and thanks to everyone for joining us. We have, I think, a terrific cast here today and a good plan. My plan is to go over that so you know what to expect for the next now about an hour and 45 minutes. Let me start by saying, if you're not already, it seems like most everyone is, but please try to keep your audio muted so we can manage the noise. We're getting a little bit of background noise here and there, but for the most part, I think doing pretty well and have a really good turnout that we're excited about. So we'll try to do our best to keep the noise manageable during the presentations in particular. Since we're a large group, we're not going to be taking time to introduce everyone. But we will be doing introductions during the breakouts. We have a small exercise where you'll be able to get to know each other. So in the meantime, if you want to rename yourself, it might make it a little bit easier for people to tell who's on the phone or who's on the line and connect to others who are interested in these issues. So I'd encourage you to right-click on your window, click rename, and enter not just the name that is your common name or familiar name, but also your affiliation or organization if you'd like to let people know who you're with, and it may stir some connections. So as Darcy explained, we've structured the 2-hour agenda to both create an opportunity to share information out to the group, learn from each other, learn from some experts in the field, but also to get more people talking and share some experience across the range of expertise from throughout all of the participants. So during the first half of the session, we'll hear from 3 guest presenters that are reporting on what we know about ocean acidification conditions in Alaska right now, how they vary around the states, and some of the areas that are more vulnerable to ocean acidification. And then during the second half of our agenda, you'll be given the option of joining one of 3 breakout groups. You'll have to choose between the Bering Sea, Gulf of Alaska, or Southeast. And in those groups, we'll be talking about how the conditions in those regions can inform some of our thinking about the priority issues that we need to be addressing as a region or as a state, what some of the needs are, unique needs of those regions, and how we can communicate best with other collaborators or decision makers, funders, other interested parties, and just generally create more of a basically sort of an environment of collaboration and coordination where there's opportunities for that. So during the plenary, we're not going to be taking comments from the audience, but you are welcome to use the chat function to track any comments or questions that you have. And we'll do our best to either address them today or at one of the future sessions or pull them into the breakouts or what have you. So don't hesitate if you'd like to use those chats, and we will be referencing them as best we can. So before we shift to our experts, let's pause because this question that Darcy posed to everybody, I think, is relevant. We want to know a little bit more about how people are feeling about the topic and the type of audience that we have. So why don't we pause and share the results, Darcy, with everybody? Maybe you are already. Is that?
I just hit share. So can everyone see that?
Great. Terrific. Okay, so it gives us a sense. So we have a range. We'll try to keep this in mind, especially during the breakouts where we'll customize some of the conversation, but it does look like over half of you have a science background or sort of are living and breathing. and about a third of the group has a general sense, want to keep track of and learn more. So we'll keep that in mind and try to target the conversation in a way that really addresses all of you in the audience. Okay, so to keep things moving and make sure that we use as much time as possible for our experts and for you to be talking, let me start with an introduction to our speakers. So as I said, we've got a series of 3 guest presenters that are going to kick off this discussion. They'll share an overview of the science that's available right now to inform our understanding of conditions and which regions are expected to change most rapidly, what we might expect in the future, which areas of Alaska are vulnerable to change. And I think we also want to just make sure that we're kind of thinking about what we don't know. So in some cases, we're going to be talking about the limits of our knowledge and understanding and how we're addressing that. So Dr. Darren Pilcher is going to kick things off for us. Hopefully you saw that in his full name and affiliation in your agenda, if you got that. But Darren is a research scientist at the Cooperative Institute for Climate, Ocean, and Ecosystem Studies. And, excuse me, pause just for a moment there, at the University of Washington and NOAA's Pacific Marine Environmental Laboratory. Darren is focused on ocean carbon cycling and the effects of climate change on marine ecosystems. And he's particularly interested in the effects of ocean acidification in Arctic and subarctic waters using computational models. Most of Darren's work is in the Bering Sea, which he's going to be addressing today, although he also has worked in the Gulf of Alaska and other regions of the country. Thank you so much, Darren, both for joining us, offering your expertise, and for being the kickoff for us today. Please feel free to share your slides.
Great, thank you, Molly. Can you see this okay?
Yep, although, yes, there we go. Now we're seeing the slide view.
Okay, cool. Yeah, so my name's Darren, and I'm gonna be, as Molly said, kicking this off with the Bering Sea. Oops. So I was going to start with a brief overview of ocean acidification, though looking at that poll, this might be a little redundant now, but just in case. Essentially, so as you might all be aware, humans have been emitting more carbon into the atmosphere. As we emit that carbon, it's accumulating as CO2 in the atmosphere and is resulting in climate change. However, not all that carbon we emit actually stays in the atmosphere. Only about 50% of it does, with the other half being taken up by the land biosphere and the ocean. And so these natural carbon sinks are providing this climate mitigation service for us, because otherwise the concentration in the atmosphere would be much larger. But for the ocean, this is coming at a cost, and that is ocean acidification. So essentially, as that CO2 dissolves in seawater, it shifts the marine carbonate system to a state of reduced pH and reduced carbonate ion concentrations, which we have data in the oceans. We've been able to observe this occurring over long periods of time. So that decrease in carbonate ion is important because many calcifying organisms use this to build their shells. So for example, pteropods, which are these tiny sea snails shown here, we've observed in the lab and actually out in parts of the global ocean that they begin to erode and dissolve at lower pH conditions. And a lot of times we use this variable called the Aragonite saturation state, which is denoted by this Greek symbol omega here, because it has this nice chemical threshold for when the shell building is favorable, so when it's above 1, versus when it's unfavorable, when it's below 1. And it provides this nice broad ecosystem level metric for kind of the water conditions. And I'll also note that it co-varies with pH. So meaning that in general, as pH decreases, that's also correlated with lower Aragonite saturation states. And we particularly care about OA in Alaska because Alaskan waters are already very cold and naturally rich in carbon, which means that it takes just a little bit extra CO2 to push them into a more corrosive state. You can see this in this figure on the left here showing the global oceans and these values of Aragonite or omega. And you'll notice that it looks actually, it's very similar to surface air temperature in a lot of ways, where the equatorial regions are much higher with it overall, whereas the polar regions are much lower in general. And then, although we have all that variability in the global oceans, we also have a lot of variability just within, say, the Bering Sea shelf itself that we've already observed. So that's shown here in this figure on the right. And a lot of this is being generated by these natural biogeochemical processes. So for instance, we've seen very low values right along the coast in nearshore waters that we think develop because of freshwater runoff. So rivers tend to be very supersaturated with carbon because they're getting all that carbon coming off from the land, from tree decaying, from leaf litter, et cetera. And so that runs off into the ocean and produces these low values of omega. Conversely, on the outer shelf, as we push offshore away from that influence, we have a lot of phytoplankton primary productivity. And so these plankton are essentially plants that they're using photosynthesis to make their energy. So that's sucking up a lot of carbon from the water and producing these high values of omega. However, those phytoplankton eventually die and sink down to the bottom. And when that happens, they get respired, and all that carbon that they had taken up gets pushed back into the water. And so it makes these very low values of omega in the bottom waters more on the outer shelf. So there's all this kind of variability going on in the background, but we do expect that because of ocean acidification, that the spatial extent, duration, and intensity of these low omega conditions is going to increase moving forward. And we care about that because Alaskan fisheries are a very important part of the state of Alaska for the economy, just for marine ecosystem services. And so previous risk assessments have highlighted that many Alaskan coastal communities are at risk from ocean acidification due to their reliance on these fisheries for subsistence and commercial use. And there's also been previous bioeconomic work related to specifically red king crab, showing that the effects of OA, we might not be able to kind of distinguish it from the natural variability due to temperature until it's kind of already too late, that we're sort of following that black line trajectory, and kind of a yield in crab is already in a huge decline. And so there's going to be a lot more discussion of the biological effects of this at subsequent discussion series talks, which I highly encourage you to attend if you're interested. But I just wanted to highlight that here since it sets the stage a little bit for why we care about this. And so in Alaska, we track ocean acidification through a number of observational assets, through, say, ships, moorings, and more recently, autonomous vehicles. And despite our best efforts with that, right, the Bering Sea shelf is a very large system, right? Darcy already mentioned the 44,000 miles of coastline. It's a very big system. It has very harsh winter weather conditions. So this is very challenging to do just with the observational data alone. And so a lot of my work takes that observational data and uses it to help develop and validate computational models of the Bering Sea for these variables. So I'm showing an example of that comparison here, where the shading in the background is the model output, and then those dots are the ship-based sampling at the same time and place. Just sort of noting general color agreement between the two, right? Especially capturing the vertical gradient from the surface to bottom that we saw in the previous figure as well. And so when we know the model is at least doing a reasonable job of simulating these conditions, right, then we can expand it out and get actually something more like the full seasonal cycle of, in this case, omega, over a particular year, so that we're moving kind of beyond just the snapshot picture that we get otherwise, and can kind of track this more long-term. So I can just let this animation play for a second. You can see that these values again get much higher during the summer as you get all that phytoplankton productivity, that photosynthesis, but then get much lower again during the winter as the waters cool and that respiration comes back up. And so I wanted to take a minute to kind of walk through this in different steps. So this figure on the top left here is just the annual surface average, this time over a much longer time frame, noting some of those same spatial features that we saw a few slides ago, where this area right inshore has relatively lower values of omega, particularly near the Yukon River outflow. Again, that river water that's rich in carbon, it's kind of driving that signal. But then these values increase as you push more offshore, and that freshwater signal goes away, and you get a little stronger primary productivity. That pattern is actually flipped at the bottom, right? So here's now the bottom waters, and in this case, now the nearshore waters are actually relatively higher than the middle and outer shelf waters, and that's because the inner shelf is just so shallow that it can't really develop that bottom water respiration of all that sinking phytoplankton being respired. That just has a much harder time developing in the shallow waters than it does in more deeper waters. But this is all the annual average so far. And so we also want to look at specific timeframes that we think are very relevant for marine ecosystems. So one that we look at is this late summer, July to September timeframe that coincides very well with a lot of the fisheries surveys that occur in the Bering Sea. And during this time frame, the surface waters are very high in omega. There's just a lot of productivity. It's just very well buffered waters overall. As opposed to at the bottom, again, the waters in the near shore are still relatively higher, but the off shelf, or not off shelf, middle and outer shelf waters are a bit lower overall again. And again, that's kind of that respiration signal developing. And the reason I'm bringing this up is because when we think about risk and OA vulnerability, sort of understanding the spatial, this variability in time and space is important to understand, what our particular marine organism that we're looking at might face, right? Like, is it in the surface? Is it in the bottom? Is there a particular time of year, a particular life stage that's most vulnerable? That'll change a lot with how vulnerable, how much risk we think there is. And so we also use this model as a tool to help us understand what the present year's conditions look like compared to a normal year. Here I'm showing kind of that same average bottom water conditions on the left, just constrained here to the US economic zone. And then on the right is how last year was different than that normal year. So sort of illustrating the waters near St. Lawrence Island and the North Bering Sea were relatively high with omega, whereas waters on the outer shelf in the southwestern Bering Sea were relatively lower overall than normal. And then we were trying to use this model output and this information to develop these indices that can kind of be like a one-stop shop, one number sort of thing for like, what are the conditions this year? And we've developed this based on so far 2 different parameters. One is still that omega equals 1 threshold. And the other is a pH value of 7.8, which has been associated with harmful effects to red king crab. And so the idea here is that it's tracking the percent of bottom water over time that has conditions less than this, than these variable, or than these thresholds. So essentially as it goes up, like it did through 2013, that's signifying water is getting more acidic. And then it's actually recovered a bit since then through 2021, sort of suggesting a little bit better conditions overall. And then lastly, so all of that has been sort of what has happened in the past. And so we also want to think towards the future, right, in terms of what are the conditions that we might expect in the Bering Sea. And so we've used the same model to project out into the future and have sort of started making these habitat, what we're calling habitat suitability plots, sort of tracking the, in this case, optimal conditions for red king crab in Bristol Bay over time. And noting that under a high emissions carbon scenario shown here on the left, that these more favorable optimal conditions erode by the end of the 21st century, and that we even get some of these extreme 7.5 values shown in the green here by the end of the century, which have been associated with high mortality of red king crab. But if we take a more moderate emissions pathway, we don't get nearly the same extent of that erosion of optimal habitat. And we also don't even get the development of those 7.5 conditions. So just sort of illustrating how it's a tangible example of how emissions reductions can benefit marine ecosystems. And I will stop there, but I'll just note that if you're interested in learning more about these future projections, we just had a paper come out that's open access, but feel free to also email me and I can provide you with a copy of that. So thank you.
Thank you, Darren, too. And if you do have a link to that paper, we're happy to put that in the chat to share with everyone. As a reminder, if you have questions, follow-up questions for Darren, you're welcome to put that in the chat. And this is a flag to sort of some of the future discussion. We will be talking more about some of the implications for individual species. So questions about impacts to species that are most of concern to you would also be relevant to put in the chat, and that will help to inform some of our planning for future sessions. So I'll move on now to introduce next Dr. Jessica Cross, who is a research oceanographer at the NOAA Pacific Marine Environmental Lab. Her current research focuses on carbon biochemistry and ocean acidification in Arctic regions, and especially along the Alaskan coast. Her main goal is to better understand how acidification processes interact with natural biochemical processes and eventually to detect geochemical and biological impacts of acidification in marine systems. She conducts research across a variety of platforms, including ship-based measurements, moorings, and mobile autonomous platforms like gliders and drones. She's very active in bringing her research to communities that can gain value from her research, as evidenced by the fact that she is on the OA Network Advisory Committee and many other groups. And she is also one of the more nimble researchers, I would say, in part evidenced by the fact that she has jumped in on very short notice to cover for Claudine, who unfortunately is ill today. Thank you so much, Jessica. It's always a bit of a challenge trying to jump into someone else's shoes on short notice, but I know that you're up to the task, and we thank you so much for your willingness.
Thanks a lot, Molly. And I want to reiterate that everything I'm about to show you today, and most of what I'm about to say, is Claudine's work. She is a researcher at the University of Alaska Fairbanks, based at the Trotheta campus, which is located in the Lower Tanana Valley, for those of you who are not familiar with that area. She is an outstanding researcher, and it is a total pleasure to be able to present this talk on her behalf today. So I wanted to get started just by sharing, Darren has already talked a little bit about the basics of ocean acidification, and I know many of the folks on this call are already going to be familiar with that too. But ocean acidification overall is a global process. I'll just reiterate that from the very beginning, and it's led to a fundamental shift in the chemistry of seawater everywhere in the world. And for the next 10 minutes, though, we're just going to focus on conditions in the Gulf of Alaska, where some regions show signs of dissolution of little sea snail shells, say that 3 times fast, likely due to ocean acidification. And what I'm showing here on this particular slide is the model output from a biogeochemical model to study ocean acidification at present and in the future that Claudine has been instrumental in developing. We use different tools to measure seawater chemistry, including shipboard measurements, moored sensors that are attached to autonomous platforms. And really briefly, it's worth noting that the Ocean Acidification Research Center started monitoring these conditions along the Seward Line and in Prince William Sound in the fall of 2008 when it comes specifically to the Gulf of Alaska. And Claudine's group took over this effort in 2018. She just received funding to also sample in the summer along parts of the Kodiak and Middleton lines for the next 10 years. So this is going to be a long-term project. And then she's also measuring conditions year-long at the Gulf of Alaska Ecosystem Observatory since 2019. This spring, she's going to deploy her newly developed carbon dioxide sea glider in the Gulf of Alaska as well. And this glider will monitor carbon dioxide, oxygen, salinity, temperature, and depth throughout the entire water column. She is planning on deploying it in winter as well to finally get a better understanding of this largely undersampled time of year. And there's a few other efforts in the community that happen less frequently. For example, NOAA conducted an ocean acidification cruise focused in 2015 and sampled transects across the Gulf, and is planning a somewhat smaller cruise in Southeast Alaska earlier this summer. There are also cruises that happen as part of the Global Ocean ship-based hydrography program. That's a worldwide program that measures transects all across the globe. And the P16 line starts in Kodiak and samples for ocean acidification all the way down to Antarctica. There's also a lot of sampling efforts in the nearshore all along the Gulf of Alaska, which will be discussed in the next round of ocean acidification discussions. Most particularly, I hope to hear Wylie talk about his sampling efforts on Alaskan ferries in the Inside Passage today. That's going to be a really fun talk. In addition to all of this observing and modeling effort that we do at various scales in the Gulf of Alaska, some scientists also monitor the well-being of organisms that are especially prone to ocean acidification. So for example, the shells of little sea snails, I'm going to trip up every time, called pteropods, dissolve in waters affected by ocean acidification. And these organisms and species really serve as indicators for ocean acidification. So even if we're not measuring OA variables and we go out and we collect these organisms and we see the dissolution in these shells, it's an easy way to check how severe the acidification is. In particular, Nina Bednarczyk identified severe dissolution of the shells of pteropods or sea snails in certain areas of the Gulf of Alaska, indicating that these areas have already reached thresholds that are harmful to certain organisms. These snails are not just an important indicator for ocean acidification, or, it's not just important that we study them because they're an indicator species, but they're also an important food source for juvenile and adult salmon. Because of the data that's been collected up until this point, we know that there are multiple inputs into the system in the Gulf of Alaska that can help control carbonate chemistry, such as freshwater discharge from tidewater glaciers, streams, and precipitation. All of these things can affect the chemistry. Circulation changes that move in water with different chemical properties can also affect ocean acidification, as can biological processes such as phytoplankton production that decrease the amount of carbon dioxide in the surface water and thereby increase pH and aragonite saturation state. However, with the data that we can obtain from these tools, we can really only get a limited spatial context and temporal context that oftentimes makes it difficult to understand these processes. This is especially true in coastal environments like the Gulf of Alaska, where there's a lot of variability and a lot of different processes that can change ocean chemistry on timescales that are a lot shorter than, broad geologic scale ocean acidification, on the order of the Industrial Revolution to now. Sometimes these are really short seasonal changes. To supplement the small data volume and the spatial coverage of that data, as well as get an understanding of how the habitat of, for example, sea snails has changed over the last few decades, we developed Claudine's group developed a high-resolution model of the Gulf of Alaska. This model consists of a grid that spans across the entire Gulf. It simulates and attempts to reconstruct the circulation and biogeochemistry from 1980 to 2013, but we're also working on, or her group is also working on, a simulation that takes that model all the way up to the present day. To do so, the model incorporates the historic data that is available, such as historic wind, precipitation, air temperature, and atmospheric CO2 concentrations, or carbon dioxide concentrations. Using those inputs, she simulates ocean circulation, temperature, salinity, nutrients, biological production, and the inorganic carbon cycle. Over the last few years, Claudine's group has been evaluating the model with available inorganic carbonate chemistry observations. And this helps them understand the model's flaws and capabilities, really similar to the way that Darren did in his presentation for the Bering Sea. So now let's take a look at the model output to see how quickly the chemical environment has changed since 1980. And a really interesting and accessible way of doing this is to see how much of the water column is still suitable for healthy pteropods or healthy sea snails. In this particular figure, we are looking at the percentage of the upper 50 meters of the water column that is still healthy for sea snails in yellow, the percentage of the water column that triggers dissolution of the shell in blue, or severe dissolution in red. And this all is across 1980 to 2013. The top plot focuses on the shelf region. And in this plot, we can see that the majority of the water column still has plenty of building blocks, although in more recent years, there are times when the healthy habitat shrinks to almost 50%. The bottom plot focuses on the gyre region, where Nita Bednarczyk found severely dissolved pteropods or sea snails. In the gyre region, the upper 50 meters of the water column were favorable for sea snail development in 1980. However, in more recent years, there were ocean acidification extreme events that turned the upper 50 meters into an area unfavorable for sea snails, which might explain the severe dissolution in sea snails that has been found in this region by Nina's group. It's important to note that these extreme events are driven by a combination of ocean acidification and natural climate variability together. During multiple years in a row, the gyre has sped up due to increased winds, which brings up more carbon dioxide from the deep waters to the upper water column, creating these extreme acidification events that are not healthy for pteropods. You need both this natural variability and the absorption of anthropogenic CO2 to cause these kinds of events. The strong gyre can also bring up water with low oxygen content, and that's what I'm showing on this slide here, leading to sort of like a compound event that is both acidified and hypoxic, and has low oxygen. So this plot shows oxygen concentration in color across the water column in the subpolar gyre from 1980 through 2013. So this is a time series, not necessarily a section plot. Dark blue means that low oxygen concentration, and red means a high oxygen concentration. So in this case, blue is bad. And hypoxia is marked by the black line. So below this black line, there is not enough oxygen for most organisms to survive. The red line throws the threshold below which it becomes dangerous for salmon, and the dotted line shows the area below which there are not enough building blocks. If we now look at about 100 meters depth with the yellow line, you can see that there are times when the oxygen is too low for the salmon, and their prey will also have issues due to ocean acidification. The key message is low oxygen and strong acidification events have become larger, longer, more intense, more frequent, and shallower over time, all exposing the Gulf of Alaska to more stress. While we've profiled a few studies with you today, we want to emphasize that the model data fields, as well as some of the observational data, are available on the AU's Gulf of Alaska data portal. Feel free to play around with any of the options there. We try to be very transparent about sharing the data that we work with as researchers, but more than anything, especially in conjunction with sessions like this, we are always interested in understanding how these can be made more useful to you. How do we help turn this data into information that can be more beneficial to you? And I hope that we'll hear about that in some of our discussion sessions later today. One of the ways that we've heard your feedback is that it's really important to study ocean acidification in a multiple stressor ecosystem context. So at the beginning of this presentation, I mentioned these these moored autonomous ecosystem observing platforms, these ecosystem observatories. And we've had the opportunity to work with one in the Chukchi Sea longer than anywhere else. These observatories collect a lot of data all at once that help us contextualize our ocean acidification measurements and also put it in context with other stressors. And this is what the data from those observatories looks like, or one piece of that data looks like. This slide shows some of the PCO2 results, or the carbon dioxide results from that particular study. Time is shown on the x-axis and PCO2 on the y-axis. In general, very high PCO2 concentrations can be damaging for the ecosystem, whereas really low concentrations aren't as much of a concern. This is a really great data series because we've been able to collect year-round data in this area where we can normally only access the Chukchi Sea during the open water season. And through these observatories, Seth Danielson and Claudine's group, as well as the rest of the researchers that work on the ecosystem observatories, have been able to spot a ton of seasonal cycles, including freshwater and mixing events, phytoplankton production, and respiration. Even things like calcium carbonate precipitation and dissolution. Like I said earlier, other moored autonomous ecosystem observatories like this one have been employed in other areas around the state, including in the Gulf of Alaska. And we really hope to be able to show you that data in the future when we get our first few years of data back. And with that, I will turn it back over to our moderators.
Thanks, Jessica. I think an extra round of applause for Jessica for such a filling in so seamlessly. Well done. And maybe we should also be applauding Claudine for obviously very well prepared presentation. And thanks to both of you for cooperating on that together.
She made it really easy.
Thank you very much. Oh, good. We've had a good exchange about how do we make use of some of these data sets in the chat. I'm going to return to that as we shift to breakouts. But first, I'm going to move quickly on to our third and final presenter, and we'll round out some of this portion of the session. Dr. Wylie Evans, thank you for joining us. Dr. Wylie Evans is a chemical oceanographer who manages the Ocean Acidification Program at the Hakai Institute, which is a nonprofit independent research organization based in British Columbia. He joined the Institute to advance nearshore OA-specific ocean observing capacity in data-poor regions of our coast. And he's studying marine carbon dioxide systems in coastal waters of Alaska and British Columbia. And as Jessica mentioned, in particular, some interesting work that he's doing in Southeast. He conducted postdoctoral research at the University of Alaska Fairbanks at the Ocean Acidification Research Center, and has also worked with some of those of you who are from the NOAA National Ocean and Atmospheric, Pacific Marine Environmental Laboratory in Seattle also. So Wylie, thank you so much for joining us.
Thank you, Molly, and thanks, Darcy, for the invitation to talk today. Thanks, Jess, for the plug, because I am going to talk about ferry work. And so, yeah, we're going to move into the complicated coastal environment. I've been tasked to talk about marine CO2 conditions in Southeast, and specifically focus on these two questions. You know, what do we know about regional conditions with respect to ocean chemistry, and where are the areas that are most sensitive to change / expected to change most rapidly? So I'm going to try and address those. But before I do that, I want to just kind of highlight, you know, what's going on in Southeast in terms of our observing effort. And the map here in the center is meant to show that. I recognize it's not completely comprehensive. I see Lauren Bell on the call, so you know, I know there's a few studies, for instance, in Sitka and Ketchikan, that might not be on this plot, but this is most of the observing effort in Southeast that I'm aware of that's happened to date. The black dots are the cruise data that Jessica mentioned, the 2015 NOAA cruise. There's been a number of cruise operations in Glacier Bay by the National Park Service. Fixed measurement locations, both with a NOAA buoy and percolator systems in Sitka and Ketchikan, a distributed array of community sampling, which are the white dots, and then the ferry track. And so it looks like we have a lot covered. But I want to say, you know, not all of this is operational at the same time. In fact, much of it isn't operational right now. Right now, really the only observing that's going on, I think, is the community efforts and probably the few things that I failed to put on this plot. And so that's important to recognize, is that, you know, our efforts change in time. And another important thing that I want to address here is that I'm going to talk about surface conditions. And most of these observations are surface water. And so a major gap in this region and probably many of the other regions is subsurface monitoring. And so I just want to highlight that for discussion as we go forward. Okay, so I'm going to just kind of give you a really quick highlight reel of a paper that was published a few weeks ago describing measurements that we've been making from the ferry, the Columbia, that many of you may have seen if you've traveled on the ferry service in Alaska. It's the flagship vessel. We've been making measurements on this vessel since 2017. I want to acknowledge AUS and University of Alaska Southeast for funding for the operations on this vessel, as well as the Tula Foundation. The link is there at the bottom of the slide. I'll put it in the chat when I'm done rambling through these slides. But let's get through it. I'm going to first orient you with the graphics here. I'm going to show you a series of very similar graphics. And so on the X and Y axis, we have latitude and longitude, and the Z axis is time. And so you're looking from really the beginning of when we started making measurements in November 2017 to when the ship stopped running in September 2019. And currently it hasn't ran since then, and we're hoping that the ship is underway again this year. The color indicates the variable, which I'll get to in a minute, but each colored line is a transect of data, basically from Bellingham, Washington up to Southeast Alaska. So we're making measurements every 2 minutes as the ship's underway. We've done 135 transits with the vessel and collected over 240,000 measurements along this 1,600 kilometer transit along our coastline. And so just to sort of walk through some of the parameters that we're measuring and what we're seeing. The first two here are temperature, sea surface temperature, and salinity. And so you see sort of the nice, seasonal warming pattern in this area. But you notice it doesn't happen evenly everywhere. And so there's certain areas that are very narrow passageways where there's a lot of tidal mixing, where we don't get large warming signal through the year. And those separate other areas that don't experience that amount of mixing, and so seasonal warming has a bigger impact. You'll notice at the bottom is the salinity, and salinity decreases in areas where we have fresh water coming in from land. And Jessica highlighted that from Claudine's talk. There's varieties of freshwater input. The largest changes in salinity are around mostly river mouths, but you'll notice this big blue sort of tongue at the top of the figure, which is around Lynn Canal and Juneau. And this is an area of glacial melt that progresses through the summer well into August and early September. And so there's a large amount of fresh water coming into that area, which is the greatest decrease in salinity along the 1,600 kilometer transit. Okay, here's where things get interesting. So this is the oxygen on the top and PCO2 on the bottom. And so as Jessica highlighted, when you have production happening in the marine environment by phytoplankton, you're producing oxygen and you're consuming CO2. And so that's what we're seeing here in this setting on a seasonal basis, is areas where oxygen is supersaturated because production is high and the CO2 content is drawn down. But you'll see it's kind of patchy, and those regions that didn't have the seasonal warming also don't have big changes in oxygen and CO2 because of the mixing that's happening. Just going to jump ahead to P8. Well, let's see, make sure I didn't miss a point here. I'm going to move this. Oh, yeah. Okay, got that. All right, I'm going to jump ahead to pH. Those areas of large production that are producing oxygen and drawing down CO2 are also accompanied by large increases in pH and more favorable saturation state conditions, which is shown here. So you have pH in those areas where CO2 is drawn down being very high, and saturation states that are favorable for shell production. But what's interesting is that I want to highlight is that area of glacial melt, and that's where things that we think covary, like Darren said, typically pH and saturation state covary in the marine environment, that actually kind of falls apart in these regions where we have glacial melt coming in, because the chemistry of that meltwater And the temperature of that meltwater is such that it is in fact corrosive, but yet the pH is fairly high, and the PCO2 is also fairly low. So it's a very unique characteristic of these coastal settings that are experiencing glacial melt. What we've done in this paper was look at sort of regional differences in the variability and the severity and the timing of most severe conditions along the transit. And so I just want to sort of highlight a few of these features for Southeast. And we're looking at the example for saturation state. And so in terms of variability, that's the left plot. I'm showing the coefficient of variation, which is the temperature. And so the higher percentage is more variable. And these areas with most variability are really on the inside waters, head of Lynn Canal, Frederick Sound, the southern end of Stevens Passage. These areas have very high variability, as opposed to areas where that tidal mixing is more prominent. Sergius Narrows on the way into Sitka, and Wrangell Passage between Wrangell and Petersburg were both regions that stood out as very low variability regions. In terms of the severity, again, Lynn Canal had very severe or low saturation state conditions, as well as that area near Petersburg and Wrangell. But a commonality here that we observed was that the nearshore waters in general, conditions were more severe. And when you moved out to the outer shelf around Sitka and in Dixon entrance, saturation state conditions were generally better. The more severe conditions were not as bad. When you look at the timing, things are also pretty interesting. In general, most severe conditions seem to occur in winter when there's mixing, but there's some oddities here. In areas where there's tidal mixing, most severe conditions typically happen in the late summer, early autumn. And then areas where fresh water are coming in can have different timing. For instance, around Petersburg and the Stikine River, the low saturation state conditions really show up earlier around June, whereas up in Lynn Canal with the melt signal that progresses so late into the summer season, it's more around August. Let's see, we saw some differences with pH that mostly around the influence of fresh water. And it's worth considering the sort of picture of variability in the terms of being able to target areas where variability is low, because not all coastal settings are highly variable, as this image shows. And so if we're really trying to target regions to diagnose long-term trends, we want to look for places where the variability is low, and this information provides a good context to do that. Okay, and finally, I want to just kind of wrap up with where we think, what we think we can tell from these data in terms of the amount of anthropogenic CO2 that is in the water and how conditions have changed and will change into the future. The top plot shows the anthropogenic CO2, which is the amount of carbon, extra carbon that's been added to the water that was highlighted by Darren and Jess in the previous talks. And what we're showing here is that it's quite variable. It's actually more variable than we thought, with very low conditions in fresh water and in areas where deep water that has lower anthropogenic CO2 than the surface water is being mixed up to the surface. One of the calculations we did was essentially take that anthropogenic CO2 content out of the, remove it from our calculations, and then slowly add it back in to the point where we could identify the first year when saturation state really became less than 1. And in Southeast Alaska, we're currently seeing wintertime conditions that are at or undersaturated with respect to Aragonite. And this calculation showed that that's a relatively new feature that's sort of developed since around the early 2000s. The current change that we've experienced over the industrial era is shown on the top here. And this is essentially the difference in what we think pre-industrial saturation states look like and what they are now. And what you can see is the warmer colors mean larger change, and that there's been larger change in the summertime relative to the winter and in these areas where there's higher production. Going ahead to the near future, which in our analysis was 2035, we can see that change intensifies. Sorry, 2035. We use 2035 because this roughly approximates when we're going to hit the 1.5 C warming level. And if society is able to limit warming to that level per the Paris Agreement preferred climate target, we can think of that as sort of the best case scenario for acidification in this setting. And so we're looking at what we think that best case scenario might be in the bottom plot in terms of saturation state. But a couple final points I want to make about pH is that we see actually larger pH and hydrogen ion concentration change in winter, as opposed to the larger saturation state changes in summer. And so that might have some biological implications for organisms that might be more sensitive to pH versus saturation state. And on average, we're seeing about a 40% change in hydrogen ion concentration in this setting, as opposed to the global average of about 30%. So larger changes in this setting. And over the next 13 years to that 1.5 C acidification level, we can expect another 17% change in hydrogen ion concentration, which is substantial, nearly half of the entire acidification signal we've experienced thus far over the last 250 years in about 13 years. So that's a striking and rapid change that we might expect. And so I'll leave it there and look forward to discussion.
Thanks so much, Wylie. This is terrific. And a big thanks to Darren and Jessica, Claudine, even though she's not here to hear it, for framing things up for us. This is terrific. Yeah, lots of clapping going on, and some good questions. I want to flag that are terrific segues into the breakouts, which we'll get to in just a minute. But I do encourage everyone to be thinking about how we talk about sharing information, and it may be that different regions, there are different organizations and different mechanisms for being able to help to get some of this information out. That is one of the main goals of this discussion series, is to make sure that the excellent research that is being done is getting out to those people that can benefit from them, and vice versa, that those people who really can benefit from research can help to shape and inform future research that is done, so that we all are learning and adapting as best we can, and just overall making best use of the investments that we're making into better understanding the conditions. So huge thanks to all three of you. And to those of you who are on the phone that have been, or on the line that have been involved in some of this research in other ways, which we know many of you have. So while we have a couple of minutes, I'm going to just take to frame up some of the breakout plans. But as a reminder, if you did have questions that were not addressed, go ahead and put them in the chat, because they will be helpful for us both in the breakouts and in future sessions. And let me just see. I was not tracking any that came in at the last minute just now, but I do want to draw attention to Andy Wall's question about making sure that we get information out to other areas of Kodiak in particular. And so we'll address that in the Gulf of Alaska breakout session. Okay, so let me tell you about how these breakouts are set up. We're going to give you, as I alluded to earlier, an option of choosing the breakout. Now, some of you may do work in multiple regions of Alaska, and so we would ask you to help to balance some of the breakouts. If you feel like you could go to any number of the regions and you end up in a breakout that has a lot of people in it, and we're kind of targeting more or less about 15 to 20 people per breakout session. If you're in one that has a whole lot more than that, you might want to flip back and go to one of the other sessions just to balance it out. Just a quick pitch to make sure that we've got some critical mass in all of the breakout discussions. So we'll move you into those three smaller groups, so more of you can be talking all at the same time. Each group will have an expert. And in this case, we'll have Wylie and Darren and Jessica. Sorry, I should say Wylie in one, Darren and Jessica in another. So Wylie is going to be in Southeast. Jessica and Darren will be in the Bering Sea, and Darcy is going to be leading the Gulf of Alaska session, which does not have Claudine in it, but we will have some other experts that are active in that area. We also will have some experts who are working in other areas of research that are targeting those regions. If you are an expert and have some additional information to provide for that region, please select that group, and we'll create some opportunities for you to be sharing some more of your work. A note on how we are also going to have a facilitator and note taker in each session. The note takers are going to record the session for documentation purposes and will help to record the ideas that are shared. Everything will be anonymous though. We will be preparing an output from this discussion series to share more widely, but we will not be attributing any of the ideas to any individuals. So all ideas are strongly encouraged, and we want to create a safe space to be throwing out new ideas and really generating a very open and trusted conversation. We also are going to give you a link to a whiteboard where you can also write. If you're more comfortable writing ideas instead of speaking, that's an alternative for you. And it is a way to hopefully have some fun and get some questions out for everybody to be thinking about together, but feel comfortable straying from the structure. If it doesn't work for your group, you can work with your facilitator and modify it as needed. All of the groups, though, are structured in the same way, and so you can adjust as you need to for your specifics. Okay, so the three questions that we're expecting to address for all of the groups. One is, how do we work better together to improve the information with local knowledge and experience from a range of stakeholders? Number two is, how can we use the science to inform the decision-making better, communicate it better, get it out to the people who know, who need to hear more about it, and inform decisions at the local, state, and national level? And then how can we guide future research? What do we need to know more about? What are some of the top priorities? And how can we encourage more research in areas that need it most? There's also an open space for any ideas. If they don't fit into those categories, feel free to throw out new ideas. So Darcy, let me, I think at this point, anything that I'm missing or any additional issues or comments that have come up that we should cover before we open the breakouts?
I think we're ready for breakouts.
Okay, so Darcy's going to post. Yep, go ahead.
Okay, so I think I just opened those rooms. Do you see, is it coming up on your screen, the option to go to the different rooms?
So if folks are seeing, it may be slightly different, but you should be able, at the bottom of the window, you should be able to have the option to join any of the groups that you see. You just need to pick one, and it'll allow you to move over into that breakout room. If anyone has any questions about how this is working, feel free to speak up if it's unclear.
Looks like folks.
Yeah, I don't see the breakout groups on my screen. If they're to be at the bottom.
Try expanding.
Yep, they're at the bottom of the.
Oh yeah, it might be hiding if your screen is kind of narrow right now. So if you expand it a bit, there'll be a little tab at the bottom of Zoom that says breakout rooms, and if you click on that, a little box should pop up. And I think, Molly, I'm going to head to the Gulf room to join the crew here. So I will be going that way.
Yep, go right ahead. I'm going to linger just for a minute while we make sure everybody knows where they're going. So does anyone not, so I've Alan, Frank, John, anyone need help? Alan, are you still not seeing the options? Frank, you need help?
I have a question. Darcy asked me to join the Gulf of Alaska group because Claudine couldn't make it. But I hope to chat with Southeast a little bit too. It looks like, I was looking at the numbers, it looks like that's kind of a smaller group. Am I allowed to come out of a breakout session and go into another one?
Yep, you can. So if she's covered, you can come back out, and I think you'll be able to switch over.
Okay.
And if it doesn't work for you, just go back into Darcy's and she'll move you around.
Okay, thank you.
No problem.
Looks like we're the first ones back.
We're the first ones back.
Where did
Oh, here we
everybody
go. All right.
Hey, Rob, you do a fair amount of outreach in Cordova. Anything you want to add since we're still in our small group, sort of?
Sure.
As everyone's coming back again, that was probably a little abrupt for some people, at least ours. We were trying to wrap up, so hopefully it wasn't too abrupt. Any reactions as people are coming? Unmute for a moment while people are coming back to center here. Any reactions? How did they go? How did the sessions go?
We had a lot of
got a thumbs
active
up from them.
text conversation. People were really like enthusiastic about using the whiteboard to like write down their ideas and really wordsmith and make sure that things sounded great. So thanks to everybody in our Bering Sea session that really leaned into using those whiteboard tools. I'm so glad that worked so well.
Excellent, good job in Bering Sea. What about Gulf of Alaska? How did you guys feel?
I'm not sure we did a whole lot of wordsmithing, but I think we got a lot of things on our whiteboard. We had way too many questions to address, so we kind of honed in on the ones that our group was most interested in, and I think that we had the specific people in the mix to address. So I think that went well.
Great. Well, a big shout out to our note taker, because Edie did a terrific job of taking notes for the group. They did a terrific first exercise, but then relied heavily on our note taker. But the conversation was active and really enriched. So I don't want to take up too much time with just the process, but it was great to have a little bit of feedback on how that worked, because of course we're going to be talking about some of our next sessions just in a moment before we wrap up, and we want to get some feedback on what worked well. So as we go through, we're going to do a quick round of reports from each of the groups on the substance. We'll keep that really high level. We will be recording these more to share some of the more detailed outcomes in the future, but feel free in the chat box to give us feedback on anything, what you thought of this first kickoff session. We're going to be doing, as you know, 3 more, and we love the feedback, and we can certainly improve on it in the future. So let me turn to, why don't we do it in the same order? Why don't I turn to Bering Sea and ask, do you have a reporter, or is anyone prepared to give us some highlights of some of the conversation that you had?
Esther, you're up.
Yes.
Yeah, I guess that's me. The people in my section are adding more ideas as we speak, which is exciting. So, okay, let's see, big summaries. So in the section about working together to improve information, our group kind of settled on a few various ideas. The main themes were that the continued or the biggest theme that emerged from that is that tighter collaboration with fishermen and industry would be really helpful there. So not just fishermen, but also skipper observations and processing workers. So sort of finding ways to incorporate the observations that fishermen and folks in the fishing industry have and pulling those into scientific reports and into scientific work. But we talked briefly about barriers associated with that, in that there's sort of the cost, the time, and then the sort of ongoing nebulous trust issues of like, okay, how do we collaborate in a way that is mutually beneficial to the scientists and the fishermen, and does not cause any problems for the fishing industry. And then moving on to science-informed decision-making, our biggest topics of conversation were, there's some feedback for sort of scientists communicating that scientists as a group need to be both better at sort of speaking directly to their audiences. So taking out technical language and really giving public presentations that meet the need of their audiences. But also that scientists need to start to feel more empowered to speak about the implications of their research and sort of stop shying away from talking about what their research actually means for the real world. In addition, there were a lot of ideas for more communication with the public, including things like more short-form lightning talks and videos that are easy to share that could maybe be shared over social media. Moving on, sorry, there are lots of other ideas on this whiteboard, so apologies for folks who are not getting called out in this moment, but moving on to the future work, we did ask the question of what are we doing well, and we did not get any responses to that, so that was concerning. But top priorities from our group were really top priorities and burning questions really focused on what are species specific vulnerabilities, what are the sort of drilling into species and variability within the year, how do we get much more specifically, how do we understand what exactly is at risk when and where, and how do we then manage for that risk? And in particular, someone brought up the point of what confounding variables might we be missing, and what sort of big ecosystem questions might we be missing. Oh, now somebody is writing something that we did well. Thank you. That was not necessary.
Thanks,
okay.
Esther.
Yeah, so that was more or less our discussion. It's ongoing, and I would be happy to. I guess I would just say now, I'd be happy to sort of write up a synopsis of this that we can send out.
Awesome. You guys, terrific job. And I'm sorry for cutting it a bit short. I want to leave time for the other two groups, but Esther, Addie, and Edie are all helping us to document some of this, and we'll be sharing it more publicly. So Addie, I'm going to quickly move on to you. Gulf of Alaska, I think, is next.
Hi. Yeah, I'll keep it brief. I think a major theme was connecting all researchers in Alaska and connecting communities and research needs before a proposal starts, to be really intentional about where the research is going and who it's benefiting. And another thing was just more collaboration between researchers, and just to reduce duplicate efforts and increase our effectiveness in a competitive funding environment. And we also talked about tools to communicate research, and a cool idea was creating a signature data set to disseminate info, and then using that same tool over time so that people could develop familiarity with the tool. And
excellent. Thank you so much.
To add that I missed.
I'm not gonna open it up because I want to leave time for Darcy to also give us a view to what's coming. Unless there's something from the Southeast group, if somebody wants to step up who didn't in the group, I will go ahead and do a quick summary. Anyone? Okay, I wasn't a good cheerleader. I didn't get someone else to do the report for us. So quickly, highlights from Southeast. I'm going to do this in order of we did a fun prioritization exercise. The top two sort of votes came, like important messages came in the form of the need for future work. One was about a lot of enthusiasm about multi-stressor research, looking at including hypoxia and warming, and how you not only do research that looks across multiple stressors, but also communicates around the very complex mix of factors. And that we not forget that there's a prevailing message that is the common enemy needs to be messaged as atmospheric CO2. That's very clear. That's the common enemy, and we need to make sure that we're reinforcing that. We also talked quite a bit about this, both the engagement, collaboration, and the messaging, and how important it was to engage communities in person. People are really feeling COVID, I think this pandemic separateness, but really talking to people, having two-way conversations, in-person conversations, long conversations about complexities to help people to really understand, and then to challenge communities who want to be involved, that need to better understand the data collection processes, and OA science overall. It's complex. We need to have those conversations in order. And there are people out there that really want to be involved, and many of you are working with them, and how important that is, and to meet them where they are, which right now apparently is TikTok and Instagram. And, like for old people like me, we got to get with it. We've got to figure out how to communicate effectively through the right tools. So terrific conversations. Thank you to everybody who was part of this experiment of how we have this in a more integrated and sort of generative way, getting more people talking at the same time. I'm going to pass it over to Darcy, since we're just about out of time, to talk a little bit about what's coming and encourage any feedback on how we can continue to make these better.
Great. Thank you, Molly. And thanks, everybody. I really like how each of those breakout groups focused on kind of slightly different things. And I think it's producing a good platform on which to build into our next two sessions, and also guide the network past this spring. So I'm going to send around one quick poll question, since you're a captive audience right now, on how you prefer to receive ocean acidification information. This can help us guide the way we circulate information in the near term, and we'll dig into this further as a group too. But I just want to first thank our speakers, Darren, Jessica, and Wylie, and our note takers, Edie, Addie, and Esther, and our facilitator, Molly, and all of you for your thoughts and ideas and your commitment to this cause. Our next session is on April 5th. It's a Tuesday. It's 2 weeks from yesterday. And the topic is on species response. What does ocean acidification mean for mariculture and subsistence? And I'm going to put a registration link in the chat, which actually I have lost from my screen. Molly, if you happen to have it handy, maybe slide it in there. And I will send you an automated Zoom link for next time. And we would also welcome any feedback on this session. I'll put my email in the chat. And we urge you, in the meantime, if you're curious about ocean acidification, if you're coming in more on the news side, to check out the Ocean Acidification Network website, which is a place that we can also continue to post all the work that you all are doing. So I will again say thanks to everybody, and we will look forward to seeing you on April 5th.
And I just got the registration link in there. Sorry for the slow response, but there you go. Anyone who wants to register for the next session. Sorry, Darcy, it took me a minute.
No, that's great. So there's the link.
I just want to thank you, Darcy, and you, Molly, as well, for helping us get all of this together. I thought this went real smooth today. So thank you so much for helping us stay organized, and I look forward to being in good hands for the next session too.
Thanks, Jess. Thanks everybody.
And Molly.
Thanks, everyone.
Thanks, everybody.
Thank you.
Molly, do you want to join a different link? And Jess, do you have time to join us also? You don't. You do. Wait, I realize you.
No pressure.
Okay. Oh, I'm still recording. I'm gonna hit stop.